All right, welcome back for our next video. So far in this first part of biochemistry, we have reviewed aqueous chemistry in the context of weak acid chemistry, in particular the effect it has on protonation of biomolecules. We looked at structure of proteins, so primary, secondary, tertiary, quaternary. We looked at enzymes and classes of enzymes and took a deep dive into chymotrypsin to get an idea of what happens in the active site, that transition state. And now we're going to really use myoglobin and hemoglobin as examples as we look at some of these allosteric effectors. So we're going to talk about how hemoglobin works and the things that affect how well it binds oxygen. So we're going to be looking, as I said, at some of these effectors. So one's BPG, proton concentration, so pH, CO2. So to kind of pick up where we left off last time, oxygen binds cooperatively to hemoglobin. So when one heme group in hemoglobin has a bound oxygen, it converts that hemoglobin. It causes a structural change that converts it to this high oxygen affinity state. So it increases the ability of the other heme groups to bind oxygen. That's kind of where we left off. So let's think a little bit about the structure and really the function of hemoglobin and myoglobin in the body. So your hemoglobin is really oxygen transport, whereas myoglobin is really oxygen storage in the tissues. So if we think about up here in the lungs, you've got a pretty high concentration of oxygen. That should be subscripted there. High concentration of O2, or a high partial pressure of O2 in the lungs, right? When that happens, that promotes hemoglobin to pick up oxygen. So we end up with this fully oxygenated hemoglobin that moves down into the tissues. And when it carries that oxygen down to the tissues, and what it's going to do is offload it to the myoglobin, which stores it there and then passes it along to the tissue. So the lower partial pressure of oxygen shifts more hemoglobin down here, and again this should be O2, down here it converts that more to the T state, so it tends to give back or give away the oxygen. Myoglobin picks it up, so we see that here. And then, oh actually that's not where we see that. We see the hemoglobin bringing it in, right, and handing it off to the myoglobin, and myoglobin's going to hand it off to the tissues. The hemoglobin in your red blood cells, it's going to now be deoxygenated and move back to the lungs. So this is kind of our picture. So now we're going to talk about why. Why does that oxygen move overall from the hemoglobin to the myoglobin in those tissues? So we're going to look at some data here. So the green line, myoglobin, pink line, hemoglobin. And you can see that at really high partial pressure, which would be about 100 torr, okay, in the lungs, that's going to be high. So that typical concentration in the lungs, both myoglobin and hemoglobin bond oxygen, have a high affinity for oxygen. If we look at the tissues, they're going to have a lower partial pressure of oxygen. And what we see from our graph is that, wow, in that difference there, the hemoglobin affinity for oxygen has dropped dramatically, in fact, 66%. Whereas the myoglobin has only dropped about 7%. So as the hemoglobin, fully oxygenated, moves to the tissues, it's moving into this area with lower partial pressure, and so it starts giving off some of those oxygens. More oxygen is going to be released by hemoglobin, but myoglobin still has a high affinity for oxygen, so as it's released, it'll be picked up by that myoglobin. This is really good for us for lots of reasons, but if you think about oxygen, so oxygen by itself is a gas, it's not terribly soluble, and we really want it bound. It's an oxidizing agent. We don't want it free, we want it bound. And so we generally want to keep it bound to either the hemoglobin or the myoglobin until it's passed into that tissue. All right, so it's passed from the hemoglobin to myoglobin because the affinity is vastly different at those partial pressures that are in the tissue. All right, then we want to look at this next graph. So when we look at this one, the pink is the one we were looking at on the last one, so hemoglobin in our red blood cells. It turns out in your red blood cells, you also have this small molecule, 2,3-bisphosphoglycerate, BPG. Okay, you can see it's 3 carbons, got this carboxyl group, these phosphates. All right, when you look at this, this is a small molecule with a lot of negative charge. When we look at our graph, if we do pure hemoglobin, if we study pure hemoglobin without that small molecule, take a look at this. It much more resembles that myoglobin curve. All right, so when you think about that, we have 2 forms of hemoglobin. You can think of it as the deoxygenated form, right, or the oxygenated form. If I can spell oxygenated. Spell it all out here. So we can have the one without oxygens bound, or we can have a molecule of hemoglobin with oxygens bound. In these allosteric curves, part of the reason that we see this change is that something is stabilizing that T state or that deoxygenated state. Okay, and what we see here is that 2,3-bisphosphate, when we lack that, there is almost no stability of that deoxygenated state, right? Every time we add oxygen, it bonds. So that tells us something about the physiological importance of this BPG molecule. So it tells us it has something to do with the affinity of oxygen for hemoglobin, and that in particular, when we have BPG, that hemoglobin oxygen affinity is lower. Okay, we see that by this shift, right, of our graph to the right. So we see that less and less, it takes a higher and higher concentration to start saturating our hemoglobin. Okay, now there's got to be a connection to the structure. So we're looking at structure-function relationship. So let's take a look at why this happens. So if you remember, in our last video, we talked a little bit about this hole or opening. We've got our 4 subunits of hemoglobin. And so over here, this little area we're showing right here. It turns out this hole is just the right size for this 2,3-BPG to fit. Okay, and if you look around the edges, we've got histidine, histidine, lysine, histidine, histidine, lysine. We have a lot of positively charged right around the edges of this opening. We have positively charged amino acids that'll be attracted to this small, highly negatively charged molecule. Opposite charges attract. And so when we form all of these attractions, that's a stabilizing effect. So when we stabilize something, we lower its energy and make it more likely to continue to exist. So we have lowered this energy, and this is for the deoxy version. So we've got our deoxy. If you remember, when we oxygenate it, we saw a change in the size of this hole, and it's no longer going to really kind of fit this molecule. Okay, it won't fit it with these specific amino acids right there in the right spots to form those attractions. So BPG is a major effector that stabilizes the T state. So when it's in there, it forms lots of attractions. Those make things more stable. So it stabilizes that deoxygenated version. Okay, there are other effectors. So this 2,3-BPG is a big effector, okay, that stabilizes the T state. So it's a negative effector. It is reducing the bonding affinity of hemoglobin for oxygen. Some other effectors we're going to look at, we're going to talk about what's called the Bohr effect. So carbon dioxide and protons produced by actively respiring tissues, right? So when you're working out or whatever, you're respiring, you're creating carbon dioxide and H+. Well, if you think about it, our function of hemoglobin is to transport and release oxygen to the tissue. BPG stabilizes hemoglobin to a degree so that at certain partial pressures, that hemoglobin will offload 66% of the oxygen. So it starts out in the lungs with almost all of it's oxygenated. It moves to the tissue, and without even worrying about the Bohr effect, it'll offload about 66% of that oxygen that'll get picked up by myoglobin. But there are times that your cells have an increased need for oxygen. So when you're actively respiring, and so we have these other effectors. When you're respiring, you are creating your oxygen, carbon dioxide, and H+. So picture you're working out, you're really working out, and you are creating a lot of carbon dioxide and H+. These are also going to be negative effectors because they're going to cause hemoglobin to release O2. So negative doesn't mean doesn't mean bad, right? Negative means that if the hemoglobin is supposed to bond oxygen, negative means it makes it less likely to bond. But that's what we want to happen in this case. We want it to give away more oxygen so those tissues can have the oxygen they need. All right, let's look at how this would happen. So you have carbon dioxide, right, forming in the tissue. Carbon dioxide, nonpolar, so it can diffuse through those membranes. It can diffuse into those red blood cells. Okay, so it can cross the walls of the tissues, the capillary, into those red blood cells. When it's in those red blood cells, it reacts with water to form carbonic acid. That's a weak acid, so it will go to equilibrium, undergo equilibrium, and what we see is it forms the bicarbonate ion right here and our proton, an H+. All right, let's take a look at some data. Think about we're forming H+, right? So the more and more H+, pH go up or down? pH goes down. We're lowering our pH. So when we look at this, we have two curves. For the effective pH, we're looking at hemoglobin bonding to oxygen at pH of 7.4 and 7.2. And we can see that a lower pH, hemoglobin has a lower affinity. This whole graph is like shifted to the right. A lower affinity for oxygen. So if we are going from our lung level of partial pressure of oxygen to our tissue level, whereas before hemoglobin would give off 66%, now it's going to give off about 77% of its oxygen. Okay, so then when we want to think about how does this happen? Why would the pH affect it? Remember that our hemoglobin is a polypeptide. It's got these polypeptide chains, and each of those amino acid residues has that R group. So it's got some R group. And as we change the pH, it's going to change the equilibrium between the protonated and deprotonated forms of some of those R groups. Okay, on the beta chain, a particular histidine is now more likely to be protonated. So we're going to have this histidine on the beta 1 chain, and now it's more likely to be protonated. We also have an aspartate here, and those will be close enough to form this attraction. So we get this new attraction that, because it's an attraction, it lowers the energy, it stabilizes it. Okay, we will also see this lysine that will now be more likely to be protonated, will form an attraction with the C-terminal. Okay, so this was one of our alpha strands and this is one of our beta strands. Okay, again, we see this attractive force that's going to stabilize this. So in the T-state conformation, these amino acids are close enough for these attractions to occur. So when pH is lowered, the T-state gets to have these extra attractions that make it even more stable. But at higher pH, okay, we're not going to see these because we won't have the appropriate protonation state. So we won't have those opposite charges forming. Okay, so the low pH allows the formation of ionic interactions that stabilize the T state of hemoglobin, enhancing oxygen release. Okay, so stabilize the T state means this stuff is less likely to bond oxygen. Let's think about the effect of carbon dioxide. All right, so the presence of carbon dioxide, right? When we look at it here, so we have pH 7.4, no carbon dioxide. That's our pink curve where we started. We have a lower pH, but still no carbon dioxide right here. And now for purple, we have a lower pH, but we also have a significant amount of carbon dioxide. And we see in each case, our graphs are shifting to the right. We're having lower and lower affinity. Okay, so the carbon dioxide is further lowering the affinity of the hemoglobin for oxygen. You can see that as we go from lung concentration of oxygen to tissue concentration, what was 66% became 77% when we lowered the pH. But now with the lower pH and the carbon dioxide, that hemoglobin is going to offload about 88% of the oxygen. So when your tissues need this oxygen, now it can be given off. So we have this carbonate groups, I can't say it today, but carbonate groups. We have the carbon dioxide reacting with the amino terminal of one of the chains. And we get this carbonate group right here, charged. That's the big piece, okay? And we get this H+. So when we look at this negative charged end of the polypeptide chain can form salt bridges. Again, it can form these attractions that stabilize the T state. So they form in the T state because of the proximity of the various ions and charged side groups. And so carbon dioxide will further stabilize the T state, making it more likely that the hemoglobin will be in the non-oxygen carrying form. All right, so as we think about the transport of carbon dioxide from tissues to lungs, over here on the left, this whole box, we've got the body tissue, right? So we got body tissue, we've got these capillaries through there, and we know that carbon dioxide produced by the tissues is moving into the cell. We get this equilibrium going, carbon dioxide and water forming our bicarbonate and H+. And we know that that bicarbonate can diffuse out of the cell. So if you think about what is happening when you have lots of carbon dioxide being made, this is going to push our equilibrium towards products. And when bicarbonate diffuses out of the cell, that'll push it even more, right? Keep it going towards products. But now these hemoglobin molecules, these red blood cells with their hemoglobin, are going to transfer or travel to the lungs. So now this whole area, we're in the lungs. And in the lungs, you're going to exhale a bunch of this carbon dioxide. So carbon dioxide can move back and forth across these membranes. So some of that carbon dioxide will come out. But you're going to then exhale it, so it can't go back in, right? We're removing some of that carbon dioxide. When we do that, we're going to start shifting our equilibrium back that way, shifting our reaction. When that happens, bicarbonate will get pulled in and used up. Every time we react to bicarbonate, we're reacting an H+, right? So we're getting rid of the CO2, and we're raising our pH, okay? Because we're using up those H+. So over on the left, we would have a greater stability because we've got that CO2 and lower pH, greater stability of the T state. On the right, we'll have a lesser stability of the T state because we don't have that CO2 and we don't, or not as much, and we don't have the lowered pH. All right, so as we talk about the Bohr effect is referring to that low pH effect and high CO2 effect. The other effector we looked at, right, was the 2,3-BPG, and that's separate. So these two are going to change concentration as they move through your body, right? Different areas have different partial pressures of CO2, different pHs. That BPG pretty much stays at the same concentration in your red blood cells. All right, so over here on the left, we have in tissues higher carbon dioxide, higher H+, so lower pH, lower PO2. We are going to see that that T state is the more stabilized one. That's the deoxy state, right? Over here on the right, in the lungs, we're going to see that that oxygenated state is going to be the one that is more stable. All right, so pause for just a second and think, what are the three factors we've talked about that stabilize the deoxy form of hemoglobin? All right, you should have come up with that. 2,3-BPG, the proton concentration, right, pH, and the pressure of CO2. All right, these slides, this slide and the next one, are really just kind of a summary. Just for your notes, they're all things we've talked about, but just to have it down in writing. All right, what we want to do before we finish talking about hemoglobin is talk about a couple interesting variants of hemoglobin. So it turns out that there are a whole lot of different forms of hemoglobin. We're going to look at three. The one we've been talking about, hemoglobin A, is normal adult hemoglobin. HbS is sickle hemoglobin. We'll look at that. And then we'll look at fetal hemoglobin. So here you have a sickled red blood cell right there as compared to our normal red blood cells. So you've probably heard of sickle cell anemia because it's a very classic example of a protein variant that has a point variation. So a point variation, just one amino acid out of hundreds is swapped out. And it turns out it's on the beta chain in the 6th position. And we see a glutamate should have been there, swapped out with a valine. So it should have been glutamate, but now it's valine if you have this mutation. And so people with this variant get these sickle-shaped red blood cells, okay, instead of our normal oval kind of disc ones. And it doesn't just change the shape, but it also becomes very rigid. They get stuck in the capillaries. It's painful. It blocks blood flow. And one of the things that causes this sickling, and we're going to look at the structure of the hemoglobin, but One of the problems that this all causes is the precipitation of the hemoglobin. So it falls out of solution. And that's because it forms polymers. So a bunch of different hemoglobins will all stick together. And so you can see kind of these rod shaped, these are polymers. These are like giant hemoglobin type molecules. And it all happens because of this one point mutation. So if we look at hemoglobin, those two places where it would normally be glutamate, that B6 position, you can see right here this blue and right here this blue. Those are those valine. They should have been glutamate. But that position is near the surface, on the surface of the hemoglobin molecule. So normally you would want it to be hydrophilic like glutamate, but it's not now. Now it's hydrophobic. And it turns out that there is another place, it's a good color to show up, right here, okay, that has this two more nonpolar residues, amino acid residues. And when hemoglobin moves to the deoxy conformation, so when it's at low O2, deoxy conformation, these two become exposed and they form kind of a cleft, like a little shape there. It's kind of like a sticky spot. And normally in normal hemoglobin, not a big deal. There's nothing really that wants to stick to it. But now we've got this other nonpolar thing right near it. Okay? And this will kind of attach and detach, attach and detach. But over time, they tend to stay stuck together. Let's look at kind of a cartoon picture. I think it'll help. On the left, you have normal, so A, normal adult hemoglobin. So we have the oxygenated version. These things in the oxygenated version are like buried in the middle, where you would expect to find nonpolar amino acid residues. But on the deoxygenated version, we see that they do come towards the surface and form these clefts that we're showing with those little blue arrows. But there's nothing to stick to them, so it doesn't really matter. But over here now, these two, now we're looking at the sickle version of hemoglobin. In the oxygenated version, again, that cleft doesn't exist, but we do see, now we've got these valines that are on the surface. And so when other molecules become deoxy, right, when we have the deoxy version and we have those clefts, now we can get attraction here. And so we see this thing polymerize into these fibers where one attaches to the other. Okay, once they stick together like that, they're no longer soluble. And so they form these fibers, damage the cells. Some people carry just one gene for HbA, normal adult hemoglobin, and one for sickle cell, and they're carriers. It's really only if you have both genes for the sickle cell that it is more of a problem. All right. Next thing we want to talk about is fetal hemoglobin. This is a great example of kind of structure-function relationship. So if you look at this graph, so the dashed line is my myoglobin. The solid line is the hemoglobin. We all have genes for fetal and adult hemoglobin, but it depends which ones are expressed. In a fetus, clearly, only one expressed is the fetal hemoglobin gene. By the time the baby's a couple months old, now really they just have adult hemoglobin, right? So there's this period of time, though, that they have that fetal hemoglobin. We've got to remember that the function of hemoglobin is to transport and deliver oxygen. And so what we want to think about is where would you expect, where would you want the affinity for that fetal hemoglobin to be? You got to think about the function, right? The function of HbF. It has to take it's still oxygen transport, but now the oxygen comes from the mother's hemoglobin, right? So the mother has the oxygen in her bloodstream and is going to transfer it to the baby. So let's come down here and look. So that myoglobin would be something like this, okay? So there was my MB. And if I look at this, I can see that the maternal red blood cells have lower oxygen affinity. We see that's shifted to the right, lower O2 affinity. Than the fetal hemoglobin. It makes sense. We need the oxygen to flow from the maternal cells, right, to the fetal cells. And then we still need the fetus to be able to offload oxygen to its myoglobin, right? So the affinity for the fetal hemoglobin needs to be between the mother, because we've got to take oxygen from the mother, and the myoglobin, because we got to be able to give oxygen to the myoglobin. All right, so two different types. One last thing we want to look at as we talk about hemoglobin is carbon monoxide poisoning. So when we look at our heme, right, we've got the four rings, we've got our histidine, our proximal and distal histidine, and carbon monoxide can come in and bind really tightly here. It's binding in the same spot as oxygen, so we would think of that as a competitive inhibitor. Normally with a competitive inhibitor, you could knock this off just by kind of overwhelming it with high oxygen concentration. So just by adding a ton of oxygen, statistically we're going to more likely replace this with the oxygen. But it turns out that carbon monoxide is especially dangerous for a couple reasons. It binds very tightly, but when it binds, just like when oxygen binds, it also increases the affinity of the other heme groups for oxygen. But it increases the affinity of the other heme groups for oxygen a lot. That might sound like a good thing, except that we need hemoglobin to not just pick up the oxygen, we need it to also be willing to give it away when it gets to the tissues. And what happens is we end up with all of our heme groups bound to either carbon monoxide or oxygen, but the ones bound to oxygen won't give it up, so it's not benefiting your tissues. It does not take a large concentration of carbon monoxide, which is one of the reasons it's so dangerous, and we have to be really careful about things like when there is a freeze with no power, you should not run a generator inside. You don't want to build up carbon monoxide in the house. All right, that was the end of our video on hemoglobin. So hemoglobin is a great example of a carrier protein, a binding protein, allosteric protein. So that was a kind of a great summary of all of that.